My name is Lisa Cohen. I'm a microarray research technician for the Robertson Coral Reef Program. I worked for Dr. Sarah Edge before she left last month for her new position, studying epigenetics at Charles University in the Czech Republic. I stutter, just to let you know what's going on. It's not because I'm cold or extremely nervous, that's just the way it is. Thank you for your patience and understanding. My presentation today is of two projects that received specialty license plate funds, one using sponges and the other with coral larvae. Corals and sponges are among the major phyla inhabiting the marine hard substrate of Florida's shallow reefs. As benthic species, corals and sponges are under the influence of changes in their environment, making them sensitive to environmental stress. Larval coral, before they settle, are also sensitive to environmental factors, both at the surface and in the water column. Settlement of the larvae on the reef, and thus the overall source of future recruitment of corals on reefs, are affected by environmental factors. The goal of these studies has been to identify biomarkers and profile gene expression to characterize and monitor sensitivities of corals and sponges to environmental change. Every organism is driven by its DNA code, found in every cell. A stressful event. Imagine an oil spill where dispersant has been applied after, or excessive freshwater runoff following a canal release, may cause genes to be expressed. Over time, after the event, mRNA is transcribed and then translated into proteins and more proteins, which can act alone or in pathways to produce metabolites. By the time proteins and metabolites are produced, the organism may start to visibly display signs of stress, such as bleaching or death. Our goal is to measure gene expression and protein expression so that we can detect responses to environmental stimuli before stress or death occur. This allows for early mitigation, potentially removing the stress. We have designed a stress-focused microarray with genes from hard corals and their symbiotic zooxanthellae. We have hand-picked genes to put on the microarray that encode for specific proteins involved in metabolic pathways turned on by toxicants, such as oil or dispersant. Other genes encode for proteins involved in aerobic metabolism, symbiosis, and reproduction, for example. mRNA is extracted from the organism, fluorescently labeled, and hybridized to this microarray. Wherever there is a spot that lights up, we know the gene is turned on in the organism. Where there is not a spot, that gene is either not present in the organism or it has not been expressed. Sponges were collected off of Fort Lauderdale with Dr. Sarah Edge, myself, Dr. Joe Lopez, and his students on an NSU vessel in May of 2012. 6 colonies of Iolocroia crassa were collected. After collections, sponges were transported to the Harbor Branch Aquaculture Facility and placed in aerated raceways with filtered seawater at 32 parts per thousand. 72 fragments were cut into equal size, acclimated for 24 hours, and then moved to 10-gallon tanks where exposures started. Experimental treatments were replicated 3 times. Weathered crude oil, which was collected in 2010 from near the Deepwater Horizon well site, was applied at 100 parts per million. Corexit 9500A dispersant was applied at 10 parts per million in the recommended dispersant to oil ratio. Sponges were exposed to environmental conditions for 40 hours and then frozen for and then preserved and frozen for future molecular analyses. Specific results and significantly expressed genes and functional groups are still being analyzed by Dr. Edge and myself, but these are example microarray images for each treatment group. If you look at the patterns of dots in each treatment group, you'll notice that they look similar between groups. But if you zoom in to an area, for example, in these red circles, there are subtle differences in some of the spots. Our computer software looks at the intensity of each spot and compares them relative to each other. An interesting result of this project is that we have successfully cross-hybridized mRNA from sponge tissue onto our microarray, which has hard coral and symbiotic zooxanthellae genes. This allows us to conclude that some of the genes on our coral microarray are conserved across phyla. Sponge mRNA samples have also been sent off for transcriptome sequencing with next-generation sequencing technology to identify novel transcript from these experiments that might not be on the microarray. The company has successfully finished processing our samples, the last we heard, and they are moving on to sequencing. Coral larvae were collected in 2006 and maintained in the facilities at the University of Miami. Larvae were collected from 3 different colonies of Porites estrioides on Little Grecian Reef in Key Largo. Batches of approximately 250 newly released larvae were immediately exposed to one of 3 treatment groups. Controls were at 35 parts per thousand, which is full-strength seawater. 2 low salinity treatments were maintained at 30 and 25 parts per thousand. Different containers were used for each treatment and each sample, and each batch of 250 larvae was considered one sample. Larvae were exposed for 24 hours and then preserved and frozen at -80 degrees Celsius until future molecular analyses. They have been sitting in our freezer ever since, until this license plate funding enabled us to analyze them. We were successfully able to extract good quality RNA out of these samples for microarray analyses and also for transcriptome sequencing. Graduate student and HBOI summer intern Ana Gonzalez analyzed these results for her summer project and master's thesis at NSU. Based on the microarray results, she found that these functional groups were differentially expressed at low salinities. I want to point out that these functional groups that are up and down regulated at 25 parts per thousand are similar to those genes differentially expressed in other studies with adult corals exposed to low salinities. Oxidative stress, cytoskeletal organization, and these other upregulated groups are basically the cells doing damage control, trying to repair themselves. We are currently developing methods to see whether results with mRNA seen on the microarray correspond to protein expression. Sometimes not all mRNA gets expressed into proteins. We have successfully developed methods to extract proteins from different species of coral, including larvae. Now that we have set up a lab to work with proteins, we are optimizing methods. We are probing with antibodies to see whether specific proteins are present, such as HeChK protein 70, a chaperone that helps other proteins stay folded properly during stress conditions, and manganese superoxide dismutase, which plays an antioxidant role. Both of these proteins are encoded for by genes found on the microarray. We are also developing qPCR as another method for verifying gene expression results on the microarray. qPCR, or real-time PCR, will amplify genes of interest and allow you to see their relative amplification as it occurs. Notice how some curves take longer than others to amplify. That means that there are more transcripts present in this sample relative to this one. Summary, we've found that our coral microarray is an applicable diagnostic tool for sponges. Salinity affects gene expression in coral larvae. We are still analyzing our results with an expected completion of these projects in June and December. Broader impacts of these projects include the future development of rapid diagnostic tools for reef resource managers and graduate and undergraduate student training. Many people and additional sources of funding have contributed to these projects. I would like to thank some of the students in our lab, in particular, Thomas Camacho and Ana Gonzalez. Ben Mason from the University of Miami collected and exposed the coral larvae. The coral microarray was developed with funds from FIO. And Drs. Voss, Pomponi, and Lopez provided technical advice.